Now, the challenge you have with the xenografts, which is basically taking things from an animal, is that inside the animal, there are things that when they come into us will react badly. But you can also have that between us, human to human, because again, even if I take something off of myself and try to transplant it later on, it can be seen as foreign. So there are ways and gradations that we have to really look at that. So just thinking that it's animal, it's completely foreign, it's completely crazy. When you track that back to the human and also self, it may not be that way.
Hello and welcome back to another episode of KU Pulse. My name is Hisham Akhtar and I'm delighted to be your moderator for what will prove to be a very interesting conversation around animal waste and the future of regenerative medicine. I'm delighted to welcome back a frequent visitor of KU Pulse, Dr. Peter Corridon. Welcome back to the show.
Hi, Hisham. Thanks for having me.
Glad to have you back. Before we get into the conversation, for those of the audience that are tuning in for the first time, what's your story, regenerative medicine? How did we get here to the podcast today?
Well, we can take it in different avenues, but I think one of the simplest ones is serendipity. So I moved here, coming up to 6 years now, or beyond 6 years actually, and I came from a typical hardcore regenerative medicine background, meaning that we did a lot of things related to transplantations. And in doing so, you had to have a great infrastructure to do that. Came here to start and build one of these. Came here 3 months later, COVID. As a result, everything kind of went haywire and we looked at slaughterhouses because we've been using that as test beds ever since. So slaughterhouses, tissues, regenerative medicine, here I am today talking to you.
Very, very interesting. So before getting into the, how the science works, let's set the stage, why animal waste? For a lot of people, the idea of using animal waste for any medical purposes might sound strange or foreign, right? So why does it work? What even begins the process of thinking about using animal waste for medicinal purposes?
Yeah. Well, that's a great question. So you're looking at why we're using these types of tissues. And looking back, the general public is not really aware, but lots of tissues that are utilized for animal studies, which actually translate into human type products or human-based studies, can be utilized from waste. Because even with the animal experimental models, you have a bunch of tissues that are not used. So looking at that and looking at the abundant amount of tissues that are available from the agri-food chain, with the growing worldwide demand for meat, I think we have a really good connection to start our conversation.
Very, very interesting. And I think it ties well into Khalifa University's contribution to UN SDGs around sustainability and repurposing, waste for responsible consumption. So we get why, or the logic behind why animal waste can be repurposed, but is there a reason why your research kind of led you to focusing on certain types of animals when it comes to the waste that could be more relevant for applications in medicine?
Great question. So looking at it in two aspects, one of the major animals you utilize as you go closer to human-based studies would be the pig. So you kind of start off with cells, then you go into smaller animals. You can look at rats, mice. Then you kind of extend it up to about rabbits, then from there, the pig, and then eventually us. Now, since we don't have a lot of pigs running around, we have to look for something that's equivalent, ergo the sheep. In addition to that, I had one colleague who had worked on camels here. He was, God rest his soul, founding faculty member as well, and also within the UAE system. And he recommended use camel. And, I thought he was always making fun of us for the stuff we're doing, but the camel has a whole bunch of different secrets because it's not easily accessible to tons and tons of diseases that we are. Camels, to be honest, should not exist physiologically. They go against so many things that we know. So trying to harness and figure out some of those things and extending them to us and relevant models, I think that's where we have a really good chance to contribute.
Interesting. I think it ties back to your point of serendipity where you never thought camels could be applicable, and yet here we are. We're obviously, at Khalifa University in the UAE. Why is the UAE an ideal place for this kind of work? Because it's quite pioneering from what, you're talking about. What supports this kind of research and potential applications as well within the region?
Okay. I look at it in different ways. So first of all, camels. We have tons of them around. And it gives us a great, quick, and simple way of developing these models. Likewise, I think I kind of look back at stuff when I was a boy growing up. So back then, I had the benefit of being in a whole bunch of afternoon programs and recognizing how the whole impetus was science, development, and technology. And where my family was in New York City, we had tons and tons of things like this. Now I kind of see here taking up that with a stronger, quicker march, because you have not just the actual work, but you have this thing advancing at the speed of AI. So it kind of takes me back, decades ago when I see so much need and so much interest, plus having, again, as I said, tons of camels just in the desert.
Was there a specific moment, during the research that, you had that breakthrough and you're like, yes, this is something that can actually have profound impact, on medicine in general? It might take a long time, but what was that aha moment for you, during that research?
So how can I say, we've had a few aha moments, but one that really stands out, and it happened actually pretty recently. We've been looking at wound healing and ECM, extracellular matrix. That's, more complicated term, but it just means everything outside where the cell is. Remodeling, fixing, like redoing your apartment. And we found a way to do that at a faster rate at what can happen at a normal, therapeutic and or human rate. So I think that was something for us is very exciting.
Very interesting. And let's, dig deeper a little bit into how the science works. So you were here last year on an episode of KU Pulse. We'll leave a link to that episode as well. If for the audience, you could just give a quick recap of where the research was, back then, and within a year, kind of where you are right now. Okay. And then we can kind of dig deeper into a complicated topic, but let's try to simplify it a little bit, for our audience.
Sure. So last time we left off looking at eyeballs and blood vessels. So extracting whole eyeballs and then secreting series of different fluids that come from them, and utilizing some of the tissues, for instance, the cornea and the blood vessels, basically trying to perfuse those to see if we can actually use these as replacement vessels in live wounds. So that's kind of where we left off. Now, moving forward to today, we've actually been able to, with colleagues of mine here, and actually students as well, they're very active in these processes and they keep me young. We've been able to extract some of those biomaterials from the actual eyeball and use that to enhance repairs in wounds.
Very interesting. Yeah, I think especially with the younger generation, just the appeal of working with eyeballs, I think brings a lot of creativity. But you mentioned wound care, and it kind of like speeds up, potentially the rate at which healing can happen. Was it difficult, to achieve this breakthrough, let's say, not from a science perspective, but, every wound is unique. Every tissue reacts differently. So talk to us a little bit, maybe, some of the challenges when it comes to wound care and how this enhances that. Okay.
Great question. So again, as you said, not all wounds are created equally. So the way in which you start these things is looking at simple wounds. So you look at, by defining it in such a way that it's the distance related to where the cells can connect is large enough such that something else has to come in to support that regeneration process. So we've recognized that we don't have to add anything in between. We just literally, when I say anything in between, anything like cells in particular, we can add a supplement which causes these cells to actually talk and migrate in a way that they generally would not do it as quickly and hopefully as homogeneously before. So that's where we have in terms of our breakthrough.
Okay. So, and please correct me if I'm wrong. So in a way, it's kind of like creating a bridge that connects the cells much quicker, because they're speaking the same language, so to speak, right? At the cellular level. Very interesting. Does it have limitations in terms of the types of wounds, that it can support or heal quicker?
Good question. So again, coming back to the fact that wounds can start off with just a scratch, or they can go into something that is chronic. So again, looking at the acute wound, we've actually done that process. Now we're getting into the chronic and long-term or potentially even end-stage wounds, which you can have, let's say, with diabetes, for instance, or major degrees of injury. One of my PhD students is actually dedicating her next 3 years to doing this. So we have colleagues in the UK who are going to help us actually make greater, dirtier mylu, if you will, within the in vitro, which is basically the simple cell model. And then as we build that up, we're going to actually start looking at something called the chick embryo model. So you can take eggs, if they're fertilized or not. You can look at windowing, making small holes in them, and you can get to see on the inside what's going on and monitor that in a live process. So if you have the fertilized egg, you have an embryo which beats, it vascularizes. So you have a quick and simple in vivo model. And again, that also relates to a bunch of things that are not fertilized in a way that could be useful or fertilized prematurely, but we can salvage that. So again, repurposing more waste. So that's where we're hoping to get to the more complex wounds and see this in vivo rather than taking it in larger, more complex animals.
Okay. Even just talking about it sounds a bit complex, but, there's this growing movement, let's say, of synthetic materials or like lab-grown human tissues and things like that. So is this process that, you're developing, repurposing animal waste, is it better than, using, let's say, lab-grown tissues? Are there limitations, challenges? Because, the argument could be made that there's only so much animal waste, that can be repurposed. So why not just synthetically develop materials? So talk to us a little bit about that debate.
Sure. So let me take it from the last and I'll bring it to the foremost or the initial point you had. So when people believe that there's not enough animal waste, there are literally tons and tons and millions and billions of tons of waste per year. So as long as we keep, indulging in our meals, there's potentially waste to be repurposed. So I don't think we're going to run out of that, such that it may even become to the point of a sustainable quality or a sustainable component, if you will.
Yeah.
So that's one aspect of it. Now, the idea of growing these things. So when we talk about lab-grown elements, synthetic versus non-synthetic, I think the debate where some people, try to get one versus the other, akin to the whole idea of the electric car versus the combustible engine, solutions lie in spectrums. They don't lie in just, one unique. So I learned this a long time ago as a recovering mathematician. Uniqueness is great, but uniqueness kind of limits us from the diverse options which we can develop this spectrum of solutions.
Okay. Recovering mathematician. Yeah. I'm sure there's a story behind that. But in terms of, let's say, the common person would be wondering that, okay, it's animal waste. Sure. The science supports that it can be repurposed. There are certain applications as well, but first and foremost, how can we ensure that it's safe?
Oh.
Right. That's, let's say, the top concern, and then maybe a couple of others that we can go through, but safety when it comes to repurposing animal waste.
Easy. I'll put this for the formation of human tissues, for example, and then we'll bring it to the animal. If you're getting a transplanted organ from a matched donor, if the organ is not in the best condition or maintained or gets to you in time, that is actual waste. So again, looking at the concept of not being too crazy or foreign, you have to sterilize them in specific ways. We can get it from surface sterilization or embedded sterilization. That's where the complexity goes. Now, the challenge you have with the xenografts, which is basically taking things from an animal, is that inside the animal, there are things that when they come into us will react badly. But you can also have that between us, human to human, because again, even if I take something off of myself and try to transplant it later on, it can be seen as foreign. So there are ways and gradations that we have to really look at that. So just thinking that it's animal it's completely foreign, it's completely crazy. When you track that back to the human and also self, it may not be that way.
Okay, interesting. And yeah, I think that simplifies it, for the general population, is because it's not so different than just doing human to human transfer. But how do you reduce the risk? Because that exists in, let's say, human transplants as well, is, rejection or immunocompromisation. So are there specific steps that have to be taken? Are there limitations as well in terms of, what we talked about earlier in applications, right? So animal waste cannot be used for this because the risk is too high. So talk to us a little bit about that.
Sure. Great questions. And kind of looking at the last point you made again, animal waste cannot be used for X, Y, or Z. As we get better with genetic manipulations, I think that last statement will go away, because even in most recent times, they transplanted a guy with a pig's kidney. And the pig actually was set up in such a way that it was actually devoid of a series of different protein expressions that would cause or enhance this rejection rate. So you see where I'm going with it. We genetically modified the pig, or basically, in a sense, maybe years to come, you may have your own sheep, for instance. Yeah. We have a Hisham sheep who has Hisham-based body parts that can potentially translate better into you, along with series of different immunosuppressants that you may have to be on to kind of help things as they go by.
So for, the common individual, there's obviously going to be a question of how the product looks, this thing of it's animal waste at the end of the day, right? So I might not get the best feeling. So talk to us a little bit about that aspect. How do we ensure that the products are adopted mainstream by making them more normal, so to speak?
That's a great question. And I have to think about this regularly, because not everyone like myself has made a career off of animal excrement or bits and pieces or gross elements, as you imagine. So we have to figure out a way in making it look indistinguishable from anything that you would have. So a vessel to a vessel, straightforward. You won't see any variation there. But even if you're thinking of, let's say, a Band-Aid, for instance, when you look at a Band-Aid, it's really a bunch of fibers and threads and or materials just sewn together, woven together. We can actually take tissues, shred them down, freeze dry them, and they can actually become fibrous-like. So they can be woven into structures that mimic or look just like Band-Aids. So hopefully give us some time and we can put them side by side, and they will look like anything maybe from a Johnson & Johnson catalog. So ergo, removing the gross aspect and making it look more mainstream. That's the hope.
Okay. I suppose, it has to be asked from an ethics perspective, right? Because I'm sure as you're developing the research and the science, is it ethical, like you mentioned, access as well for the population and so on. So what are your thoughts around that debate, let's say, when it comes to animal waste? That's one thing, being available, but then, like you mentioned, personalized waste, availability. Your thoughts around that?
Sure. Great questions. And I'll take it back from when I was a grad student. When I was a grad student, there was PETA. I don't know if you know, not to be mistaken with me, but PETA, the organization that is against animal testing whatsoever. And we could not go out and discuss and tell people things because, again, worry of PETA. Now you have those fears and those misguided views because it's not like you are trying to hurt animals as you go by. So that's where your ethics comes in. Mm-hmm. Anything that has to go into the human body, there's a process. You want to make sure that before it gets into Hisham, it's clear and clean and not going to actually reduce you from the population. So I think necessary steps help. And a lot of the things that we're using in terms of this waste is to devise a series of test beds, which basically enhance the way in which we can do these things. High throughput, make it faster, quicker, far cheaper, because again, it's waste. And limit the amount of in vivo type work we do before we literally get into something that can be used for you. So along that, think of me as someone trying to outline spectra, not just clear single solutions. Does that help?
Yeah, definitely. So I think, the need of the hour as the science progresses is going to be regulations, kind of, as is with a lot of the medical industry, right? So are we seeing, those regulations being developed already? For example, the processes of, like you mentioned, sterilizing and making sure it's safe before it even reaches clinical trials, let's say. Are those processes developed? Or is that something that's kind of like, as the science increases, then the regulations will come along as well? Because it's always a chicken and an egg situation, right? You don't want to over-regulate to stop the science, but you also don't want it to be, we'll figure it out later.
Exactly. So I guess in that case, we look at it from the concept of the cool thing about life is we have so many chicken and eggs that are here simultaneously. So nothing is really coming from ground zero. Years and years ago, the FDA has been one of the pioneers in outlining what we should and should not do with humans. And again, they've been evolving. They've been looking at fast-track methods to get things done. So again, as we go by, we're seeing more chickens basically producing better eggs, and better eggs spurring on more chickens. So you have this kind of back and forth going. So I think it's a really interesting time for regulations and the advancement of the science. So maybe in the next 20, 30 years, who knows? You can see more of these things available.
Yeah. I'm not sure if 20, 30 years, people have the patience anymore, given that we want everything now. But, we've talked about why the science works, how it works, and all of these things. I think it would be very interesting if we can personalize a little bit, to our audience. So if there's some, story that you can share of actual, let's say, not research, but implementation, that was done, or something really cool. We brought eyeballs and then we kind of went away.
Okay.
So maybe around that, if there's something you can share with the audience.
So one thing that always, I tell my students this a lot, it's always near and dear to my heart. So the research lab that I can say that, made me starting off into this major field, one of our pioneers back in the day, well, still is one of the great minds in the field, Dr. Atala. He had one student that he actually performed one of the first transplants of the materials that we were building back in the day, back in the day. Mm-hmm. And this kid was so, how can I say, inspired by this. He went on to medical school because of his own story. And then he came back into our lab and I actually trained him for a bit. And we would always joke about this. We were like, if it wasn't for you, and we point at his scar and stuff, we wouldn't get to this point. So having him now being in a position where he is a natural medical practitioner who understands this process intricately, he is a really good advocate for this and hopefully can spur more of this type of science on when, us eventually namaste, retire, and forge the other groups to come in.
I mean, he's quite literally, a walking example of why the science works.
Yeah.
So I think that's quite interesting. Okay, so we talked about applications within wound care and speeding up the healing process. As you're developing the research, what other areas do you think this can go? And even if we haven't figured out the how yet, because as is with any science, there's never really one application, right? So where do you see this kind of evolving into?
Okay. So I mentioned loosely last time, you can look at the idea of cultivated meat, because again, it's all the same basic body parts and starting products. You basically take it and reverse engineer it and hopefully try to make that into a delicious steak. So that's one application. I think also other things that might come about, the use of these things is, and again, I have a friend who is more synthetic. He's interested in wood-based applications and glues and so forth. So he's thinking that, we can utilize some of these things for other types of products, like adhesives, which in my mind, I was never interested in, but I was like, maybe we can actually do that. Yeah. So I think those two you will see up and coming.
Okay. Yeah. Interesting. Yeah. I mean, I have to ask, how would a glue or any synthetic, potentially be made from animal waste?
So for these things, so what really is a glue? A glue has the ability to form really tight bonds. That's really it. In our body, we have things that form really tight bonds naturally.
Okay.
So ergo, tight bonds.
Very interesting. Yeah. So it's like kind of not looking at the product itself, but kind of what makes up the product.
Exactly.
Yeah, very interesting. Okay, so we've had a very interesting conversation around the science, why it works, how it works. Broadly speaking, about regenerative medicine, you're an expert in the field. Where do you see the industry going? Because at Khalifa University, we're all about innovation and pioneering a lot of things. So regenerative medicine, is there a future where we can just grow things based on the science? What mysteries are, making you curious about the future?
Well, I get curious and sometimes sad and sometimes excited, depending on the day and how the experiment goes.
Depending on your calendar for the day.
Exactly. But I must say that I'm also generally a very hopeful person. So this is just part of my character. And I think it extends in lots of the work that I see as well. Because it's not just here. You have people across the globe, really, really smart people who are, how can I say, just livid to get this thing done. So again, within the next 20 to 30 years, as I said, and I always like to say that time frame because I know that we want things like yesterday. But if you get something like yesterday that you're going to put into your body, you may not want it tomorrow.
Yeah.
So I always caution, give us some time to get these things properly. Even in, these fast-paced situations, it's not worth it. Mm-hmm. So I think in 20 to 30 years, you'll have far more growth of lab-based cultures, lab-based tissues, lab-based implantables, lab-based wearables, lab-based dressings, and potentially even lab-based delicacies. So that's what I'm looking forward to.
So everything from kind of potentially what you eat to what will fix, the impact of what you eat.
Or even stick, the glues. Don't forget the glues.
Glues. Very, very, very unique application, I must say. We could have this conversation for like another couple of hours, but we do have to wrap up. Before we do, one message, one takeaway for our audience, assuming they didn't understand anything about the podcast, or what we talked about today, what would you say to our audience?
I always mention this to our students. Okay, just because it's complicated doesn't mean that's impossible. And we'll bring it back down to something that anybody can recognize. Rihanna. Listen to the immortal words of Rihanna. Work, 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 work. Just go in the lab and work. And eventually something will come up.
Yeah. Dr. Peter, thank you so much for joining us again. As we mentioned at the start, Dr. Peter has been on KU Pulse a few times. We will link, previous episodes below. Regenerative medicine is quite an interesting topic, as we talked about. However, patience, needs to be there, even if the curiosity is high, because there can be serious consequences if we don't get it right. Dr. Peter, thank you so much for joining us. If you're interested in more science-based content in an interesting way, please follow us on Spotify, YouTube, or SoundCloud. I'm Hisham Akhtar. Thank you for being with us today.